Can everyone see my slides?
We can.
Okay, thanks. Yeah, so I am currently doing a postdoc at the Department of Biomedical Informatics at Harvard Medical School. I'm working with Professor Sumaiya. I'm also affiliated with Professor Vikram Khurana's lab at the Brigham and Women's Hospital. I'm also an associate researcher at the Broad Institute of MIT and Harvard, and I have to declare my competing financial interest that I'm a consultant for a biotech startup, the Capo Brain Sciences. So let's get started. So before we dive into any of my work, I will just like to give a brief overview on what is computational biology. So we know that computer science is a field that gives us all of these tools as our arsenal, so we can develop algorithms, we can do mathematical modeling, pattern recognition, machine learning, statistics, and we have methods for doing privacy and security, simulations, high performance computing, etc. Now, what does it have to do with life sciences? We can actually employ all these tools to understand different biological systems. And that is the gist of computational biology. Like, we can understand from the microscopic biomolecules to organelles, to cells, tissues, organs, to like the whole organisms and the population of organisms, and also the entire ecosystem and the biosphere. Is there a question?
Please mute yourself if you are not speaking.
All the participants must mute it.
Okay, so yeah, so now it's not just an applied field where you take the tools from computer science and apply it on biological problems as is, because biological problems and data come with its own challenges, and you actually have to develop novel methods to solve those problems. And computer science can play a huge role in that. In fact, if you are familiar with the certain, like you all probably heard about deep learning models because there's a lot of hype in recent years. And last year there was a news, and this is news flash, and you see it's an artificial intelligence news, but it has solved one of the biggest problems in biology. Although this is a news article, so it's a bit exaggerated, that like DeepMind's AlphaFold AI program predicted 3D structure of every known protein. They did not do it for every known protein, but they did it for many, many proteins that was not that was not solved before, basically. So we can actually do very cool things using computer science in life sciences. Now, let's take a step back and just understand the basics of molecular biology, because I know in BUIT, we did not have any courses related to biology, and engineers sort of shy away from biology and they don't want to study biology, but I'll keep it very basic, okay? So we all know that the heart of a cell is the nucleus, right? From our high school biology, and nucleus contains the chromosomes. Right, humans have 23 pairs of chromosomes they inherited from their parents, and then the chromosomes contain the DNA, that is the building block of life, right? So DNA is a 3 billion base pair sequence of these 4 characters, A, C, T, and G. That's what we need to know, and there are parts of DNA, segments of DNA, which we call genes, that contain functional information. Now, these functional or coding blocks of DNA, they are transcribed to messenger RNA or mRNA. These are single-stranded sequences of, again, 4 characters that are adenine, uracil, cytosine, and guanine. And you probably have heard of messenger RNAs during COVID, because the vaccines we are getting from Pfizer and Moderna, those are mRNA vaccines. Now, these mRNAs are translated to proteins, right, which are the functional units of every living organism in the planet, right? So these mRNAs encode the information for proteins, and then proteins are expressed to give rise to different function, different phenotype. By phenotype, I mean characteristics of a species, like eye color in humans, or like your height, your BMI, your weight, etc. So this is basically the central dogma of molecular biology. Now, thanks to technological advances, we can study each of these levels, molecular levels, at unprecedented detail now. So when we study the DNA, the study is called genomics, and when we study the mRNA, the study is called transcriptomics, and when we study protein, it's called proteomics. But the biomedical data is not confined to these three areas. In fact, we have many other omes, right? So even though all of the cells in our body contain the same DNA, same blueprint, but cells differentiate into many different things, right? We have blood cells, we have muscle tissue, we have different organs, but the blueprint is the same. So how does it happen? So epigenome is the chemical modifiers that actually control these processes, like what a cell would differentiate to. And then, so there is the study of those chemical modifications called epigenomics. Then also our genes and proteins and mRNAs interact with different small molecules that are present in our tissues and organs. They're called metabolites, and the study of those metabolites is called metabolomics. And then we have many different microorganisms, like bacteria, viruses, funguses, living inside our body, on the surface of our body, and also in our environment. And they also control different biological pathways and functions related to us. So their study is called metagenomics. And together, all these omes control different biological pathways and functions that give rise to different phenotypic variability, that like different characteristics in humans, and also disease is a phenotype, right? So if you have a disease, you can have many different characteristics within different individuals. Certain people have more severe disease, certain people have less severe disease, and certain people are healthy. So one of the goals of computational biology is how we can use the data from all of these different omes to understand the molecular basis of a phenotype, a characteristic of a particular species, right? So now I will switch to how it all started for me. So as Sadia mentioned, I did my undergrad in computer science and engineering in the Department of CSE in BUET. So we have to do a thesis in the 4th year. And initially, I started doing my thesis with Dr. Masood Hassan. He is a theoretical computer scientist. He is now teaching at a university in Medina. He's no longer with BUET. But so when we went to Professor Hassan for thesis, so he introduced this problem to us, which is called protein folding problem. You probably remember the example I showed for our AlphaFold 2. So the problem is very basic. Like the proteins are represented as sequences of amino acids. So there are 20 amino acids. So it's a 20-letter alphabet, and you get like sequences of characters like strings. We call them strings. So given the sequence of amino acids for a protein, the problem is to computationally determine its 3D structure. Right? This problem has been plaguing scientists for more than 50 years. And so in 1972, the Nobel laureate in chemistry, Dr. Ang Finsen, actually postulated in his congratulatory speech that this sequence of amino acids for a protein can fully determine the 3D structure. Now people have to solve it. Now, going from this sequence to structure, actually in nature, proteins go through thousands, like innumerable conformations before it's settling down to minimum energy conformation like this. This looks like a jumble of beta sheets and alpha helices, and you can have many, many possible configurations to choose from. So the search space is humongous, right? So this is not an easy problem. So when we have a very difficult problem, mathematicians and physicists and biologists also make simplifying assumptions. So we started studying this problem in a very simpler setting, like where each amino acid can be characterized into two categories. It's called an HP model, so hydrophobic, hydrophilic model, where some amino acids are polar and some amino acids are non-polar. You can think of it as a blue and red dot, basically. So we are reducing a 20-letter alphabet to a 2-letter alphabet. So it's a much simpler string, and and this string needs to be folded in the 2D or 3D lattices. So it's a square lattice and a cubic lattice I'm showing here. And the idea is you do a walk of this string such that the red dots are adjacent to each other, right? So sounds very simple, but turns out that this problem is actually very difficult. In fact, this is a result from 2009 by Bonnie Berger and Tom Layden, which shows that protein folding in HP model, the simpler model, is also NP complete. So those of you who don't know, NP complete means a class of computational class of problems that are so difficult that they are computationally intractable. So when a problem is like that, you try to find finding optimal solution is hard, but you can find a near optimal solution using approximation algorithms. So we were studying some approximation algorithms for 2D HP lattice models, but then Dr. Hassan had to go for a sabbatical. So we actually switched to a new supervisor, Dr. Sohail Rahman, who is an expert in stringology. So stringology has nothing to do with string theory in physics, but it's study of computational methods to analyze string data. String means character sequences, right? Now, biological molecules are represented as character sequences, as I mentioned before. DNA, RNA, and protein all have character sequences. DNA and RNA use 4-letter alphabets, and proteins use this 20-letter alphabet, right? And biology loves repeats, like nature loves repeats. So it repeats characters and shorter sequences of characters within the larger sequence. So, and if you have repeats in strings, then they can be efficiently represented by data structures called cover arrays, where you represent these repeat information using integers. And you can store them using in a very efficient format in the computers. So it has to do with string compression and other things, right? So then Dr. Rahman introduced us to this paper that solved this problem. Like, if you are given a sequence of characters, can we find the minimal and maximal cover arrays efficiently? So this Crotchmore and colleagues found linear time solution for that, but they introduced an open problem in that paper that we solved this problem for going from a string to a cover array. But if you are given the cover array, can you reconstruct the string with certain properties efficiently? Right. So Dr. Rahman wanted us to solve this problem in the thesis, which has not been solved before. So in our thesis, we actually could solve this problem. Like, given a cover array, we could construct a string with minimum alphabet in linear time, and we gave an algorithm for that, and we proved the correctness and everything. And we presented this paper in Qualcomm conference, and later it was published in the Journal of Discrete Mathematics Algorithms and Applications. Now, we also solved a similar problem where we looked at another data structure, which is called a border array, that contains the prefix and suffix information of a character string. And the idea was, can we construct a degenerate string where certain characters in the string are missing and can assume any character possible within the alphabet? And we can do it using both unbounded and minimum alphabet letters. And this was also published in a Journal of Discrete Algorithms. So at this point, this was purely theoretical research. We were developing algorithms, we were analyzing runtime and complexity, and so on and so forth. Now, the switch for me happened from theoretical CS to computational biology when I went for graduate studies at MIT. So after graduating from BUET, I briefly taught at BUET for 1.5 years. And during that time, I applied for a Fulbright fellowship, a Fulbright Science and Technology fellowship, which was very prestigious. And unfortunately, I was from the last cohort of Fulbright. And after that, they closed that program. But yeah, so Fulbright actually allows you to apply to multiple schools in the US. I applied to 7 schools through Fulbright, but MIT was not one of them. I applied to MIT separately. But then once I got admission into MIT, I told them that I have Fulbright, so I had some additional financial support. Now, at MIT, I joined Professor Bonnie Berger. If you remember, she was the one who proved that protein folding problem in HP model is NP complete. And we were housed in this Stata Center, this crazy looking building, which houses the MIT Computer Science and Artificial Intelligence Laboratory, which is known as CSAIL. Now here, I had a lot of independence in choosing what problems I wanted to solve. And Professor Berger always encouraged us to come up with a vision, like, why do I want to do the work I'm doing? And what problems are most exciting to me. And so then I wanted to see the application of the CS algorithms that I know and solve some actual problems that have some tangible impact, right? Because theoretical CS algorithms, you prove correctness, you do the runtime analysis of an algorithm, you probably make an algorithm slightly better, but it doesn't have any tangible impact on your surroundings, right? So then I started thinking, and I was most excited about understanding diseases, complex diseases in humans. Now, I view diseases in a very simple, through a very simple lens. Like, there are 2 types of diseases in the world. One for which causes are known and preventable, so they can be prevented or they can be cured. And the other types of disease are causes for which causes are unknown, and we can only treat the symptoms. Unfortunately, the set of diseases for which causes are unknown is huge compared to the ones for which we know the cause. And the overreaching goal of my research is, can we make the set of diseases for which the causes are unknown smaller and transfer them to the known disease set? Okay. So then how can we do that? We have to obviously mine biomedical data. Now, there are a lot of challenges in mining biomedical data. The first is multimodality. So I showed that there are different omes, and you have genome, you have transcriptome, you have proteome, epigenome, and so on and so forth. And each of those omes give you a somewhat different picture. But so those data types are correlated, and some data types are orthogonal. And a single story from one ome is never complete, right? So we have to look at many different modalities of data, and we have to come up with models that can efficiently integrate these different types of data. Then there is disease interconnectivity. So humans never get one single disease, right? If you look at aging population, they have many different diseases simultaneously going in them together. And also different diseases share symptoms, right? So diseases share symptoms, and diseases co-occur in patients, so which increases additional complexity when we are trying to understand one disease. And then there is lack of generalizability. So unfortunately, most of the biomedical data focuses on white European patients. And so there is lack of representation from non-European ancestry and minority groups. So most of the methods that have been developed, most of the drugs that have been developed work really well on the white European population, but they don't produce results that are generalizable to other ancestries or minority groups. So whenever you have challenges there are opportunities, right? So if we have multimodal data, we can come up with efficient integrative representation learning. If we have disease interconnectivity, we can come up with methods that can analyze diseases together. And if there is lack of generalizability, we can design generalizable methods. Right? So I have worked in each of these areas. So in my master's, I developed a method that integrated data from transcriptome and proteome and many other and from literature to identify genes and modules for autism spectrum disorder. It's a neuropsychiatric disease that happens, a neurodevelopmental disease that happens in very early age children, and it's quite common in the population. So we presented this work in ISMB 2015, and then I went on to do more work in my Ph.D. So we tackled the problem of disease interconnectivity. So we developed a 3-tiered integrative transcriptomic pipeline for autism and all of the other diseases that co-occur in autism patients. So this was the first large-scale study of autism and its co-occurring diseases in human genetics, and this was well received, and we published it in Genome Biology in 2016. Then we switched gear a little bit and we moved to metagenomics. So metagenomics is the study of microbes like the bacteria, viruses, etc., that inhabit our human body. And unfortunately, we do not know a lot about the microbiome, but it's a treasure trove of data. And actually, your microbiome is contains unique signatures that can identify you, and it can tell very useful information about your health and disease. So we developed a method called Carnelian that is very robust and accurate in telling us what the microbes in your microbial sample doing in your body, and that information can tell us about what a healthy microbiome constitutes of, and when a disease occurs in a patient, what changes in the microbiome, right? And we presented this work at ECCV, and also later it was published in Genome Biology, right? So then I moved to do the postdoc at Harvard Medical School in Brigham. So when I was finishing up my PhD, so these set of excellent PIs, Professor Shamil Sunayev, Bikram Khurana, Jian Peng, and Anthony Cooper, they together got a big grant from the Australian Parkinson's Mission to study the genetics of different synucleinopathies. So synucleinopathies are a group of neurodegenerative diseases that are characterized by abnormal aggregation of a protein called alpha-synuclein. So by abnormal aggregation, I mean the misfolding of alpha-synuclein protein. As you see that protein folding, why it's important? Because when proteins are folded correctly, they function correctly. When proteins misfold, they can cause many problems and many diseases, right? So a little more background on these synucleinopathy diseases. So classical alpha-synucleinopathies include Parkinson's disease, dementia with Lewy bodies, multiple systems atrophy. And so all of these diseases are characterized by inclusions of alpha-synuclein. So you form these kind of fibrils in your neurons in Parkinson's and dementia with Lewy bodies. So they are called Lewy bodies, and they form in dopaminergic neurons. So for Parkinson's, your substantia nigra is affected. For DLB, it's more diffused, and your neocortex and substantia nigra are affected. And for MSA, you get gliocortical inclusions, and they affect your brainstem and other things. So basically, they are diseases of brains where your neuron certain types of neurons slowly dies, and it's a disease of aging. So as people get older, they get these diseases, and their brain starts shrinking, and you get lots of problems related to the movement and cognition, right? So this set of excellent PIs, they scouted me to study the genetics of this problem. And I was very excited about the opportunity because I get to work with computer scientists, statistical geneticists, and stem cell biologists. So this was a great opportunity for me to work on. Now, the diseases we are focusing on, they are relatively rare in the population. So previously, our problem was huge data, right? We were looking at many different types of data, multimodal data, interconnected diseases, and lots of data from microbes. But now our problem is we are looking at relatively rare diseases in the population, which affects 1 to 2% of the population, like Parkinson's. DLB affects 0.1% of the population, and MSA affects around 0.0006% of the population. So it's very rare now, but all of these diseases are debilitating. So it's very important to study them. So now our challenge is small sample size or insufficient data, because even though we have made tremendous advances in sequencing genomes, because these diseases are rare, we simply cannot sequence enough patients. Now, there was a study in Nature Genetics in 2021, which says that to understand a complex disease like Alzheimer's, for example, and to explain 50% of the heritability, that means how these disease transfers from generation to generation, you need to sequence at least 10 million samples. But the current data sets we have for Parkinson's disease contains only a few hundred samples. Okay, so and we are not going to get like millions of samples anytime soon, and we cannot really expect that to happen because we don't want people to get diseases, right? So what we can do, we have to, because we are challenged with small sample size, we need to design powerful methods for mining small data sets, right? The other challenge is heterogeneity in samples. So I previously mentioned that people have different symptoms, right? So when these Parkinson's patients come to clinic to see the doctors observe that these patients have very different symptoms and they are very different from each other. It seems like it's not a single disease, but a collection of diseases, right? So for example, like when you measure, evaluate Parkinson's patients, you use this Unified Parkinson's Disease Rating Scale or UPDR scores to sort of measure every year, like how bad they have become compared to the previous year. And if we plot these scores for a certain group of patients, then you can see that some patients progress very slowly over the years, and some patients become bad like really fast. Now, why understanding this heterogeneity is important? Because when you are treating these patients, you probably cannot give them the same treatment, because then it will not work on the patients that are becoming worse progressively, right? And when you are doing randomized controlled trial for a patient, if you don't consider patient heterogeneity and mix them in the treatment phase, then what happens that after the trial is finished and you compare them, then there is no visible difference, because you are putting a person who is very fast progressing with a very slow progressing patients, because this is a randomized controlled trial and you have no way to tell who is who. So can we identify which patient is going to become very worse very early and which patient is going to become worse over a long period of time, so they can handle the disease better? So then for clinical trials, we actually want to look at only the patients who are going to deteriorate very fast. And that will help us identify drugs that work, because then we can isolate those patients in different subgroups, and the faster progressors in one subgroup and slower progressors in others. And then we can actually see which fast progressor subgroup are deteriorating, responding to the drug versus who are not responding to the drug, right? So that helps us design better clinical trials. Now, these are the challenges, and how do we do that, right? So as I told that the team I work with, they contain both stem cell biologists who work on biological assays in model systems, right? Yeast, fly, or human iPS cells. And they study the protein alpha-synuclein in detail, which other proteins it interact with, and it can find interconnections and give us a network of proteins or modules in which interact in these model systems. But they cannot tell us if this protein will do the same in humans. So then I also work with human geneticists and statistical geneticists, where we develop methods to look at large population scale data. And we can actually take that network, the biologist isolated, and include that in our mathematical models to increase our power to detect if there is any mutations or variations in the genome of those patients that isolate them and give rise to a certain phenotype in them that can explain their disease or their progression, right? So then basically the positive genetic findings from our approach provides a validation for the network, and also it helps the biologists to refine their search space, and they can look at less and less number of proteins in more deeply. Okay, so we call this iterative refinement approach, where we bridge two different fields in order to gain insights into a particular disease. Now, so my current research focuses on solving the addressing the challenges of insufficient data and patient heterogeneity, right? So I have developed a method called RBTT, which identifies whether there is increasing burden of rare missense mutations in pathways involved in disease risk and severity. So that method first came out in our medRxiv preprint with Tam and Julie, and then we also published a paper in Cell showing that our method can be applied to link a certain group of proteins that are interactors of alpha-synuclein with risk of Parkinson's disease. So this was the Cell paper that came out last year. And then we are developing another method that addresses the limitations of previous method and helps us better identify like mutations in different genes and gene networks that are associated with disease risk, progression, and also different phenotypes. So that is forthcoming. And then we are developing another method that uses diffusion-based models and unsupervised clustering to identify patient subgroups in the patient population that is already rare. And we can identify gene programs that are responsible for making some people making the disease progress faster in some subgroup of patients compared to the others, so that we can target them better in clinical trials, right? So far, any questions? Did I go too fast? Like, was it hard to understand? Okay. I'll take that everyone is still with me. So now I will give a brief example of how computational approaches can solve biological problems, and I'll talk a little bit about one of my projects. Are there any questions so far?
We have a question.
Sure.
If you would like to talk about it now, or we can wait till the end for our Q&A session.
Either is fine with me.
Okay, then we can go now.
Okay.
So the question is, how do you quantify placebo effect?
So basically, I told you that we measure like certain things, right? Where to measure like how the patient is progressing, right? So when we are doing the trials, we know like who is given the placebo and who is given the treatment, right? And then basically in the placebo group, some people might still fare very well and some people may degrade, but we have the measures, right? So who is doing well, who is doing bad, and so we can actually quantify like how much they improved versus how at a certain time point. So this is a time series, so they are evaluated frequently. So clinical trials can be 3 months to 2 years, and they are evaluated frequently at different time points. So you basically compare them with some baseline to see how much they improved, how much they not. And then if they receive the treatment, that will be the treatment effect. If they receive the placebo, it will be the placebo effect. Does that answer your question?
This question was from Asif. I hope you have the clarification. Asif, are you there? I think yes. This is
Okay.
good to go. Thank
Okay. Okay. Any other question? Okay. So
there's one more question.
Okay.
From Ridwan. So can freezing gate biomarker help in the clustering?
I did not understand the question. It is
So Ridwan, can you please unmute yourself and explain the question?
Yeah, hi. Very nice one. So I wanted to know whether there's biomarkers for Parkinson's, right? So the external, like using EEG, like more like seeing the behavioral patterns, like Parkinson's have freezing gait patterns, and that also leads to some kind of biomarkers in the brain, which can also be fed into your system in order to do the unsupervised clustering with this very less number of data, more clarified, or let's say gives more information to get more, get other ideas about this disease. What do you think about that? So
yeah,
it's like,
definitely. Yeah. So those measurements you were saying, because the idea of clustering is that we want to cluster patients early in the disease, right? So we can measure those things in patients and we can use different modalities of data, right? So I primarily focus on molecular data, so genomic, transcriptomic, etc. But definitely those other measurements that come from the patients, like gait measurements, and like they measure different CSF levels and everything. So they can help in clustering, but sometimes they can also introduce noise, because if a patient is on medication, that can alter those data measurements. So it happens quite frequently. So as long as you can get feed clean data to the clustering algorithm, we can definitely probably get some clusters that are meaningful.
So I also have another question, and it's very interesting. That's why I'm asking this question. So when you mention about the protein folding, and when it goes into a 3D structure, and we're still binarizing the problem into philic and phobic, but the thing is, then there is no solution. There wouldn't be enough solution for it. So it would always remain a problem that cannot be solved. So are you still into those aspects, or have you gone into the broader perspective? Are you sometimes do you focus on the internal structural patterns that can arise from it? Does it pique your interest even now, or no?
So the HP model is a simpler model, right? You probably can, even if you don't find the optimal conformation, you can find near optimal conformations using different approximations. So those are within a certain multiplier or scaling factor of the optimal solution. But I don't personally work on protein folding problem, but now we have deep learning models like AlphaFold 2, which came up with this amazing architecture, like the evolutionary transformers, where you can actually feed into the sequence information in the form of multiple sequence alignment, and also the protein geometry information to actually infer the 3D structure. And there have been a lot of work done on this problem. So deep learning is another beast. So it can do many, many computations that traditional algorithms cannot do. And
Sure.
now we have like sort of solved structures for most of the proteins out there.
So a question in this regards is like, so when we do like deep learning, which I also do sometimes, but the thing is, when we do that, we rely upon an enormous amount of data in order to get it done. And it's luckily this protein structures, we can feed as many data as we can. But is any like scientist thinking about the counter theoretical product that is coming out of these deep learning infrastructures, which is so complex that people cannot comprehend, but are they trying to find out some analytical solutions of this deep learning architectures that they're just trying to see in an empirical fashion?
So, I mean, there is a whole research field to understand the theory behind deep learning, because this is deep learning came out from engineering solutions, right? So you have a problem, you have access to like huge resources at these large tech companies, you engineer the hell out of a problem and then you solve it. But we don't understand the theory very well. So what people are trying to do now, they look at the intermediate features that these deep learning models learn and then try to dissect those to do to interpret what is actually learned by the model, right? So there is a whole research field, and people are trying to design interpretable deep learning models based on that. But we have some intuition, like how protein structure works. So we actually intuitively know what the model is doing, right? So it tries to basically, from multiple sequence alignments, these models try to understand how different amino acids co-evolve. Then it tries to understand like which pairs of amino acids should be closer in space. So it learns long-range dependencies from the paired representation information. And then using these information, it first tries to infer the backbone of the protein, and then it tries to identify the other components that hang from the backbone and tries to identify the angles. So that gives you the minimum energy conformation of the protein.
Thank you.
Yeah, sure. Okay. Yeah, how are we doing on time? Like, should I continue or should we open the floor for Q&A?
So maybe you can use last 3 minutes to finish your presentation as much as you can. Then last 10 minutes will be the question answer.
So 3 minutes will not be enough for this problem, but yeah, I can introduce the problem a little bit. So maybe this slide will be relevant. So a classic problem in computational biology is we take the DNA, we do DNA sequencing, but as I mentioned, DNA is a sequence of 3 billion base pairs, right? So it's a 3 billion characters long string. We cannot read it from end to end. So what the sequencer does is it breaks down the DNA sequence into sets of short reads of, say, they can be 100 to 200 base pairs to 1 kilo base pairs for long reads. And then, so basically, and there is redundancy so that every position in the DNA sequence is covered multiple times. So then what are biologists going to do with these set of reads? So you get a huge bag of reads of, like, say, gigabytes of data. Now, how do we know what is the original DNA sequence that produced it? So we have some reference genomes for humans. So, and then the idea is we map these reads to the reference genome. So there is a mapping problem, and this is a computational problem, right? And so basically what is this doing? These is taking the shorter sequence of reads, and it is aligning that to the longer human reference genome to reconstruct the genome that was being sequenced, right? So then why do we want to reconstruct the genome? Because we want to identify positions in the genome that vary compared to the reference genome, and those are called mutations. So that is done by a process called variant calling, right? And we can do, so when we sequence a group of individuals who have disease and a group of healthy individuals, our goal is to compare variants from patients and healthy individuals to understand the molecular basis of the disease process. So that's like classic human genetics. Now, we can actually make, so here we are dealing with one species that is human, and we want to understand healthy versus disease. Now we can make this problem even more challenging. So we can go to metagenomics, where now you don't have one genome, because your microbiome contains many different bacteria and viruses. So instead of shredding one books, you are now shredding probably a library of books. And the output of this sequencing is that you get this shredded paper pieces, and your task is which book is of, can we identify each of fragment is coming from which book, and which topic each fragment belongs to. So the topic question is the function question, and the book question, which book question, is the taxonomy question, right? So that means that which read is coming from which bacteria, and which read is coming, mapping to which protein. Now, this is just one microbiome. So if we are wanting to do comparative metagenomics, then we essentially shred multiple libraries, right? So we get terabytes of data from different libraries, and we want to see, like build profiles, like you have libraries on one as rows and books as columns, then you want to quantify like how many copies of each book is present in which library, or if you do it the functional way, then for each library, you look at the topics, which topic contains how many books in each library, and things like that, right? So, and this helps us compare disease versus healthy microbiome. Then also we can do metagenomic biomarker discovery and clustering analysis and ecological modeling and whatnot. But the idea is, because your microbiome, which can be studied from your poop, and that's very easy to study, but definitely not an easy computational problem to solve. But it is definitely, poop collection is very easier than blood draws or other things, right? So our goal is to build accurate quantitative profiles from these microbial samples and derive consistent generalizable conclusions. Okay. So should I go into the algorithm? Okay, I'll quickly go over that. We'll skip the motivation. So the good news for us is there is a lot of functional redundancy in microbiomes. So if we compare microbial function from people from different parts of the world, we can actually get generalizable results. But that doesn't happen with taxonomy, because different species of bacteria inhabits different parts of the world, even different parts of your body. So looking at taxonomy is not a wise thing to do. Now, basically the core problem is we are given metagenomic reads, how we can find functions and build comparable statistics. Now, by functions, biology means many different things. But in our case, we will look at basically which reads correspond to which enzymes, because they drive metabolic function for different bacteria. Now, we give two solutions in this pipeline. So reads are not directly mappable to proteins, so we have to do some translation. That's not where we will go in today, but our contribution was we do alignment-free gapped k-mer binning, so to map these reads to functions, and then we do some statistical normalization to make these functional profiles comparable. So I will talk a little bit about this alignment-free gapped k-mer binning. So the intuition behind is like high sequence homology means functional similarity. So if two proteins have similar sequences, they do similar things. And but if two proteins do similar things, that doesn't necessarily mean that they will have high sequence similarity. So if you try to match sequences perfectly, that will not always give you good classification if you are trying to identify function. Basically, that's the takeaway. And so I'll skip these things. So functional binning problem can be compared to a multi-class text classification problem, if you're familiar with that. That means that if you are given multiple documents and you have to identify different topics that are present, and there could be unknown topics in the document that the classifier has not seen before, right? So the process in text classification is very simple. We take the document, we do shingling. That's basically shredding these documents into pieces of small sequences of characters. These are sets of k-mers. So these documents become very high dimensional points, right? So a document, depending on its length, can have many million k-mers in it, right? So then we provide do something called min hash and locality sensitive hashing. So hashing means helps us. It's a computational technique to just bin different things together, right? Put in separate bins. Like, for example, if I want to bin people, I can say that we have 12 bins corresponding to 12 months in the year, and people who have birthdays in the same month can go into a bin. So my function is birthdays in the same month, and so that's the hash function, right? And locality sensitive hashing is basically puts things that are similar in the same bucket. Basically, that's the idea. So then now your dimension becomes number of bins, right? So these short similarity preserving signatures correspond to each bin, and that's a low dimensional space. So we are doing a dimensionality reduction here. And then we train a classifier based on these low dimensional points to identify function, right? So that's the idea. And any questions up to this point?
Not at this point.
Okay, yeah, so I'll quickly gloss over things. So usually when you do locality sensitive hashing, people choose these hash functions randomly, right? So then the problem is then each point within the k-mer is not covered well, but our unique contribution in my PhD thesis was we do specific type of locality sensitive hashing called Opel-Gallagher hashing that ensures that every position within the k-mer is well covered. So this gives us a better performance in classification. And these Opel-Gallagher coding actually come from the signal processing. So it's Opel and Gallagher gave this idea of low density parity check codes to introduce redundancy when information is being transmitted over the network. And this technique was introduced first by Yunan Luo and colleagues in 2018 for biological sequences. And then we used this technique in functional classification, basically. So that was the idea. So I will probably stop there, and I will go to the acknowledgement slide. And because, yeah, I think we don't have enough time. But yeah, over the years, I got to work with amazing mentors, like Bonnie, Shamil, Vic, Jen, and Anthony. And I also had a ton of collaborators in all of my projects. And I have been generously funded by Fulbright, Ludwig Center, Garvin, Center for Microbiome, and also NIH. Thank you, everyone, for listening.
Thank you so much, Sumaiya. That was a lot of information, and that was really exciting to hear all this from you. So we can take one or two more questions at this point. So if anyone have any questions, they can unmute themselves and ask the question.
Hello,
Yeah.
Sumaiya, Dr. Sumaiya. Thanks for giving us time. Fabia, I appreciate your time. I wanted to understand a little bit about where Bangladesh research community in computational biology stands. Maybe you have much more knowledge than us. I'm not in this field, so my question might be a little superficial. So can you first comment on that? Like, is there any prominent scientists working in Bangladesh in computational biology, and what kind of resource you think Bangladesh research community needs to excel in this field?
So There are a lot of people in Bangladesh who are working on this. So I mentioned Dr. Sohail Rahman, who is still in Buet, and then some of our colleagues in CSE department, like Dr. Shamsudjoha Baizid, Dr. Atif Rahman, they all work in computational biology, and they are professors at Buet. And so there is a good research community there, and people are doing computational biology research in undergrad, and they're regularly going to grad schools like at MIT, CMU, and other top schools doing computational biology. And so there is a Slack channel where we maintain connections between mostly Buetians, but outside Buet, I think there are several other universities. I think the Shaha group, from Shejuti Shaha and her father, they also do good work, commendable work in genetics. And then Dhaka University Genetics Department, they have a thriving community. And there is, I think Dhaka University Microbiology has good community, and ICD-DRB has very good research going on. They also collaborate with Broad, MIT, Harvard. So very high quality research is coming from that place as well. In terms of resources, I guess it always helps when people from the US collaborate with people in Bangladesh, because that helps get the work greater visibility. So that's something that could be considered. And I think also giving students opportunities to collaborate while they're still in Buet will help their future careers.
Thank you.
Yeah. Thank you, Asif. That was a great question. Any more question at this point? We have a feedback from Maliha Sheikh. She said it was really detailed. Thank you.
Thank you, if you enjoyed it.
Thank you, Maliha.
I have a question.
Go ahead.
So Sumaiya, so you have you graduated from MIT, you did a master's and PhD from there. So any suggestion for undergrad student or master's student here in USA or in Bangladesh who want to go to top schools, like how they can enrich their profiles? So any suggestion for them?
So I'm not sure like what they actually look for in a student when they take the students, but they definitely want to see unique applications. How passionate you are about the field, what kind of problems you are most interested in, how relevant they are, and are you thinking in the right direction? I think that's something they look for in the students, and also they look at the preparation of the student, like how the student, if he or she is consistent in their academic careers, are they hardworking? Are they achieving things? Are they learning from their failures and things like that? So maintaining a consistent academic performance is important. Then also doing exciting research, right? So they definitely want to take people who has the aptitude and ability to do research and probably succeed. So that can be seen from the publications that you do in undergrad. It's not a prerequisite, but it definitely helps the application. That's my understanding. And also recommendation is very important. So they definitely want to see recommendation letters from people they know. I think that helps a lot. So if you're doing research with someone who is known to the community outside of Bangladesh, then that helps a lot.
Right. Yeah. Thank you. Thank you for the
Thank you, Mahmoud. Thank you, Sumaiya, as well. I think we are running out of time.
Can I get just one question over here? I think it would be valuable for everyone. So
yeah, go ahead.
Oh yeah. Sumaiya, I think what happened, I mean, this might be something that is coming from my point of view. It might be wrong as well, but what I'm trying to say is, like, I think you solved a problem which I guess Dr. Bonnie had an issue in the first place, and you targeted that problem because there was a professor in Buet who guided you in that direction, and that's what basically opened up the gates for you to explore all of these things, and that's how the ecosystem really developed for you to go over there. What I'm trying to say is that this ecosystem in Bangladesh, no matter which university you're from, is a commonplace. It's very difficult to replicate this kind of ecosystem. So do you have any plans in order to, make this kind of opportunity that you have got, and also the direction that you have gone? Let's say the problem was minimized for you. Like, how can you do this minimization problem for everyone to explore MIT and other universities in a much more, I mean, the frequency becoming larger day by day. So I think that's what I got to understand from your journey.
Yeah, so I definitely was fortunate because the problem that was introduced to me at the beginning of my thesis was actually interesting to me. So it depends on the mentors you get, right? So it's a hit or miss. Like if you get good mentors, like who can actually understand where your interest lies and then helps you choose the problem that you work on, that's great. But it doesn't have to be at the undergrad, right? So undergrad, we were developing algorithms and proving runtime complexities and stuff like that. But then the problems I worked on in my PhD are very different. And by the way, the problem that Bonnie proved that is NP-complete is still NP-complete. We did not solve this problem. And AlphaFold is from Google's DeepMind. So I did not work on that problem. But the problems I worked on are very different, and not even the problems that Bonnie usually works on. So she gave me a lot of freedom to choose the problems I want to solve, right? So yeah, but yeah, definitely undergrad research experience is more like understanding the research process, right? Like what research entails. I think as long as the student gets some set of some idea about that, that should be fine, because PhD is where you really explore research and find your niche.
Thanks.
Thank you, Sumaiya. We have 2 more feedbacks from Faizul. Great presentation. Thanks, Sumaiya. And from Avi. Great job, Sumaiya. Fantastic presentation. Thank everyone. Thank you for joining.
Yeah, thank you for listening.
That was very interesting topic to know about. At this time, I would like to hand this over to our President Tanzima Mustarin to say something. Tanzima, are you here?
Here. Hopefully you can hear me.
Yes, we can hear you.
Yeah, okay, sorry, I had some technical difficulties in the beginning, but I joined and I was able to listen to the entire presentation. So thank you so much, Sumaiya. This was very detailed, very informational. Actually took me back to my high school days. That was a good, a good memory to go back to. So that was really nice. I really, it was detailed, and as everyone has been saying in the chat, I really enjoyed it, and hopefully, they'll get some eye-opening, situations from this, from this presentation. To the rest of the team, again, thank you for joining us. We do, we do this once every month webinar. So hopefully you can also join us next month, for the next one. As you can see, we started off this year with a bang, right? Sumaiya's presentation was certainly, a very good one to begin with. So thanks again, Sumaiya, for spending your cold Saturday morning with us and enlightening every one of us here. So hope to see you all next month for our next webinar. Hopefully everyone can join. Enjoy the rest of your Saturday and weekend. Thank you for coming.
Thank you, everyone.
Thank you, everyone.
Thank you, everyone. Have a great weekend. Bye.
Bye-bye.